room as well. Okay, so I'm based at Imperial College in London. So for those of you who have been to London, we are right next door to Paddington Station. So we have 3 trees, slightly different to the beautiful campus that you have here in Viçosa, to say the least. And St Mary's is famous for 2 things. One is that it's where Sir Alexander Fleming discovered penicillin. And secondly, and I know that Downton Abbey is really big in Brazil. I've never seen an episode of Downton Abbey, so I don't know what it's all about, but the historical links, but all the royal, or pretty much all the royal births take place at St Mary's. So that's the other thing it's famous for. Okay, so what I'm going to do today is talk about some of our work and give you some relevance, some messages as we go along, and I've split it into 2. So the first one is to do with diagnostics for serotyping for Actinobacillus pleuropneumoniae. That is a massive mouthful, so I'm going to refer to that as APP all the way through. And I'll tell you about background to APP for those that don't know the organism. So this is my favorite organism. I love talking about this organism, so it's nice to be here. That's importance of serotyping and then the development of these diagnostic PCRs. And then part 2, I'm going to change slightly to a more complex point-of-care devices for not just APP, but many pathogens of the porcine respiratory disease complex. So that's slightly different and how it's worked. And what I'm going to do is really tell you a story about how these both developed. Yeah, and you think this will take maybe a year or so, but actually both these projects are years and years of work. Okay, let's go. So one message before we start, or put diagnostics into the context. So we live in a world where AMR is very much, antimicrobial resistance is very much in the agenda. And so this talk could have been given in the AMR side of things as well. So to reduce or eliminate bacterial pathogens, there are lots of ways or lots of methods that you really need to come together, I think. And diagnostics is one of those. So here we have vaccines. That is the best way. If you can prevent infection, that's great. Antibiotics, reduction in antibiotics. We heard a bit about that yesterday. Very important for the AMR agenda to bring that down. Environmental factors, so things like biosecurity, crowding, etc., in terms of pigs. And diagnostics is just a part of this. Yeah, it's an aid to help this. It's not the be-all and end-all, it's a help. But it can be a big help. And I hope to show you that the diagnostics that we're developing actually can make a difference. Okay, so let's start with the multiplex PCRs for APP serotyping. So I am hoping everybody in the room knows what a PCR is. So let's differentiate out the 2. Singlex PCR, single PCR. So that's where you have 2 primers and you're targeting 1 gene or 1 bit of DNA. That's a single plex. So multiplex is where you have many primers in your single tube and you're targeting many different genes. So that's the multiplex. So the advantage of a multiplex is that you actually get much more data from your money. 1 single reaction rather than, say, 20 reactions, individual reactions. So that will hopefully, for those that don't know that terminology and the students, that should now be clear. Okay, so APP, it's economically important pathogen across the world in every pig sector. So when you look at the data to say, or papers that have been published, how important is this, you see a bit of variation. So I've seen from about £4, so that's 20 real, up to something like about 180 real per pig in terms of, so it varies widely, different production systems. So it can be very significant in terms of economics. And basically, you can put it into subclinical, which is here, where you have no clinical signs or lesions at slaughter. And these are a massive problem because you're not impossible to detect, but they're really hard to detect. And these guys here, they can pass on APP to other pigs and cause mayhem. This is the one that's the most found across the world, which is chronic. So low mortality, few clinical signs, reduced growth rate, and you get lesions at slaughter. So this is a lovely picture here. There's some from my friend Andrew Rycroft, and you can see the big leap. Well, you can't see very well, I think, at the back, the big fibrous lesions here. And what also can happen is you get acute disease. And this really happens when you get subclinical pigs here. You can't detect they've got APP. They go into a naive herd, and then next morning, you've lost 70% of your herd who are dead. So this is devastating for the farmers. Okay, I won't do that one. So a few facts about APP, because this will make clear why we're doing the serotyping. So it's got 19 serovars. These are based on capsule. And here's a beautiful picture from Mario Jacks, now retired, was in Montreal. And you can see on this outside here, staining in ruthenium red, you get a huge amount of capsule. Yeah, so sugar coat. Then epidemiologically, you get biovar 1, so they require NAD for growth. This is because they have a truncated NADV gene. Yeah, they need NAD for growth in the lab, or in the pig as well. And biovar 2, NAD independent. They have a full-length NADV gene. And then you have the toxins. These are the most important virulence factors of the whole lot. So you have the APX1 to 3 toxins, which are hemolytic and cytotoxic, and these are what cause the lesions. And then you have a molecule which is called APX4, which has sequence motifs which suggest that it's a toxin. I don't know of any paper which has shown toxin activity for APX4, not one. And so it's called a toxin because of these DNA sequence motifs, but there's no proof it's a toxin. Yeah? So I think it gets mislabeled in the literature. But it's a very useful molecule because APX4 is only found in APP. It is not found in any other bacterium. So you can use it as a marker. It's brilliant. And so what you'll see in the literature is that serovar and toxin production, APX1 to 3 production, is related. Well, it is, mostly, but there are quite a lot of exceptions. And so where you have APX1 and 2, that's called high virulent strains. So these are in red: 1, 5, 9, 11, and 16. Those are the high virulent strains. And here at the end, we have APX4, which is in all of them. So these are the serotypes, serovars down here, and the ones in red, they are the nasty ones. They're high virulence. Where you have a single APX toxin of 1, 2, or 3 produced, they are low virulence, normally. There are exceptions. So there is a link between serovar and APX1 to 3 production. And it varies from country to country. So this is data from us, from the UK, and you look at the serovars in the country, and we have 2, 3, 6, 7, 8, and 12. And this is 1995 to 2007. and pretty much 80% are Siravar 8, and up until 2015, actually up until now, Siravar 8 is the one that causes a problem in the UK. And Brazil and the UK are twinned here because Brazil has a lot of Siravar 8. Not many countries do. So for the UK, it doesn't change very much. But for places like Korea, United States, it has changed dramatically over the years. So Korea has moved from Siravar 1 to Siravar 5. Yep, there's very little Siravar 1 in Korea now. Completely changed. And that's the normal situation, actually. Country to country, it changes within the country. Oh, there we go. Okay, so I've introduced the Siravar, I've introduced APX4 in particular. So why do we need to know the Siravar? What is the diagnostic potential of knowing the Siravar? And it's pretty straightforward, actually. So the vaccines that are most used are whole cells. They're inactivated with formaldehyde, and then they're given. The problem is that in most cases, whatever Siravar you use to make the vaccine will only protect against that Siravar. There are exceptions. 3, 6, and 8 cross-protection, 4, 11 cross-protection. So if your Siravar changes in your country, you actually need to know what it is to use the right vaccines. And so that's an important driver. And you also want to know, like Korea, when new strains come in, particularly the high virulence ones. So in the UK, in England, Wales, and Scotland in the UK, not Northern Ireland, we have only had one report of a high virulence strain, Siravar 9. I tried to get that strain. It died in the freezer of the people that reported it. I'm not convinced, I'll be perfectly honest. Not convinced. But in Ireland, so that's across geography across the water, they have Siravar 1 and 5s. So they have high virulence Siravars. And I don't understand to this day why England, Wales, Scotland does not have Siravar 1 and 5. It's only across the water. It is very close. And there are transport of pigs. I do not know. But it's very important for us to monitor, because if we get Siravar 1 and 5 in England, Wales, and Scotland, then we're going to have to do things quite differently. Yep. So that's why. And I've told you that it changes, so you need to keep your serotyping diagnostics up to date. And the people that do the serotyping here are veterinary diagnostic laboratories. So that means that when you develop these tests, you have to talk to the veterinary diagnostic laboratories about what they want. And what they do is isolate the APP from the lungs, and then they do the serotyping. So what they want is a test that is fit for use in the lab. You could do this as a point of care, but is it worth it? Answer no. You want to know whether you've got APP1 first, and then secondly, what the serotype is. But if you've got APP, you might want to give antibiotics. So then you'll worry about the serotype afterwards. So this is the way that the industry have told us. Oh, there we go. No, wrong one. There we go. Okay, so what's the starting point for this? So many years ago now, we did develop a multiplex PCR, which was based on some published ones already. And our contribution, which is not clear from this paper, is actually that we didn't have anything for Siravar 3 particularly, which we have in the UK. So this was our starting point. We'd already designed this multiplex PCR to pick up 1 to 3. There were already ones available for 5 to 8 and 10 to 12. So trying to hit 3 was important for us here, because that was important for the UK. we were being very UK-centric. It was important to us. So this was our starting point. So this was in 2014. And just to point something else out here, so you'll see in every one of these I show you that you have the APX4 amplicon. And this does two things. One, it tells you your PCR's worked. Normally, you have controls. But two, it tells you that whatever your isolate is, it's APP. It's absolutely specific. There's no other bacterium which will give you a positive result. So that's good for us. Okay, so that's where we started. Oh. And what we didn't have in this this multiplex PCR is we didn't have ones for 4, 9, 11, 13, 14, and 15. So 9 and 11, they're the high virulence ones, so we definitely wanted those. And at the time, 15, that was originally isolated in Australia, but was starting to move, especially through Southeast Asia. Yeah, so we wanted to include that as well. So, so the question for us was, could we extend this particular one, only 15 serovars known at the time, to cover everything? Yeah, so it could be used worldwide, actually, was try and fit for the whole of the pig industry worldwide. So this was our first thought. So what we did, what we thought we would do was extend the one that I've just shown you, first PCR, PCR1, to include 4, 9, 11, and 15. And these are the strains which are mainly BioVar1. Yeah? And objective 2, have another PCR to include 13, 14, which are BioVar2 normally, and also confirm the BioVar status, like with BioVar2 status. And we've achieved this by whole genome sequencing of known and missing serovars to formulate new PCRs. So let's go away and do a whole load of whole genome sequencing. And when we just started this, there was a paper that came out from Hungary, from Laszlo and Rita here, and they described a new serovar, serovar 16, so we actually got involved with them and developed a single-plex PCR here for serovar 16. So that was when, just after we'd started, actually. Okay, so it's to cut quite a, well, 2 years' work, probably, maybe more than that, 2.5 years' work in one slide. We sequenced over 220 APP strains from mainly the UK, but also across the world as well through collaborators. So there's a message here for students these days. Collaboration is good. Yeah. If you want, if you look at the statistics, collaborations between countries, papers tend to be of higher impact. Yeah, that's what the statistics say. So for your career, those of you that stay in academia or even industry, I think there's a very strong message there. And so these are all the serovar 8s here, a lot more because they're UK. And here are our missing ones, serovar 4, 13, 9, 11, 15, 14. And the other thing that we did was we included isolates in people's collections and our own, which had been classified as non-typable before. And it led to the identification of 2 new serovars, 17 and 18. These were sitting in, one in our freezer and one in a freezer of somebody else. Yeah. So we identified 2 new serovars by this whole genome sequencing. This tells you about the power of whole genome sequencing. We could have gone straight into this test, tried to do these tests without whole genome sequencing, but we decided actually the best way to do this, to get the best data, was to do whole genome sequencing, and that has paid off. And so it's based on capsules. And the way that capsule loci work in APP, also gram-negative bacteria, is you have about a 20 kilobase region that goes across here, and you have the capsule export genes, which are highly conserved, not only in APP, but across gram-negative. And you have the capsule biosynthesis genes, which is where it's all different. And this is where our PCRs are based on. Ooh, let's go back. So, what's our current strategy? Is to have a multiplex PCR1, which gives you 1 to 12, 15 plus APX4. And if you have an isolate, and it's APX positive, no other bands, then you might go on to multiplex PCR2, 13, 14, 16 to 18, NADV, that's BioVar, and APX4 as a check. So that's, this is what, I don't know how many years, 3 years to develop 2 PCRs. So, great. So I've talked about 9/11 together. So it turns out that the capsule loci of 9 and 11 in the biosynthetic regions are very similar. Yep, there is, it's one base pair difference, one base deletion in Siravar 11. Oops. So we took the decision to make 9 and 11, the primers for 9 and 11, so they would have the same size. So you're going, okay, what about the industry? Well, the industry agrees with us. Because you've got the capsule is so similar, 9/11, this doesn't make any difference to herd management. None at all. If you have 9/11, you could use 9 or 11 as a vaccine. It wouldn't make any difference. You would still get good cross protection. So it doesn't make any difference. So clinically, it was absolutely fine to do this. And we obviously checked with diagnostic labs, etc. And we've also used this to find some very strange, unusual strains across the world. Let's just have a look at this one. So this is Canada, Spain, Canada again, Denmark. So if you just look at this fourth lane, you've got Siravar 2 here, and you've got BioVar 2. They are incredibly rare strains. I think I only know of about 6 of those worldwide. I don't know what the significance evolutionary of that is, not sure, but it's a brilliant epidemiological marker for these strains, if you've got them. If you've got Siravar 2, BioVar 2. And we eventually came up with a comprehensive 1 to 18 here, and that is out there and it's being used. And we've also had to modify it slightly for a Siravar 19 strain. Again, this is because we had something in the collection and another strain came into us which was non-typable. We went through all this and said, I think this is a new Siravar. We had the whole genome sequence done and it was a new Siravar. And then we have to change some of the primers for our PCRs. But this is about keeping your diagnostics up to date. You have to keep them up to date for your different countries. The beauty of, I think, of what we've developed is actually, it doesn't matter what country you're in. At the moment, this covers all known Siravars. And then we were contacted by actually a company. So this is a company we've had a lot to do, which was CEAD. And they said to us, well, we're doing this worldwide Siravar study, but can you use your technique? So instead of having to get the isolate, which send it to the lab, they streak it out, take a colony, normally one colony, can you do this from clinical material? Yeah. So we said, okay, let's give it a go. And so what we used was FTA cards. So for those of you who don't know what FTA cards are, these are like little bits of like Whatman paper, filter paper, and they have some reagents in them, and I do not know what they are. I tried to look for the patent, but the way these work is that they break up viruses and bacteria, and the DNA sticks to the paper. Yeah, and you can transport this in the post, so cross countries, whatever, and that's fine because they're non-infectious. And if you look at the protocols for FDA cards, they say to get the DNA off, or nucleic acids off, is that you have to use salt and extraction. So what we found was that all you have to do is add London tap water to these and boil them for a couple of minutes, and the DNA comes off just fine. What I don't know is that's telling us about London tap water. There is a lot of chlorine in London tap water, but it works fine. You don't have to spend an hour getting the DNA off. It just comes off beautifully. So here's an example here. So these are from pigs in Portugal, actually, which were, these samples came from Portugal, sent through the post to the UK. Let's have a look. Okay, well. Okay, so what we have here is the multiplex PCR done off the FDA card, and here we have extracted genomic DNA. And it's sample, so I may just point at these. So on this side, you've got Siravar, hang on a moment. So here we are, so these straight off the FDA cards, you've got some Siravar 4, Siravar 5, Siravar 8. These are in lung samples. The FDA, they're like little dots. You push them on the lung sample. That's all you do, nothing else. Yeah? You can open up the lesions and just push them in. This is just fluid. So it's very straightforward. And here you have the same results on extracted genomic DNA. Okay, so what was interesting to us here was we had a number of pigs where they had 2 Siravars in their lung samples. And this is shown here. So here you have L20, this is Siravar 5, the reference strain. Here is our pig lung sample, P3, and it's got Siravar 5 in it. And it's also got Siravar 17 in it. Yeah. So diagnostically, these would be normally missed. You plate out, you take a single colony, and you do the AP serotyping. Here, you've got the serotypes, both of them, in the same sample. So you go, okay, so what are the implications? Well, if you have problems on that farm, then, and you streak out and you get one colony, you make an autogenous vaccine, you make a vaccine from that strain, actually, your APP won't go away because you've got another serotype. And it may explain why there have been so many reports of failed autogenous vaccines. Yeah? So now you can tell. So this works beautifully. We've done a lot of this. Okay, so here we are. Here's our current one. Start at the top. Multiplex PCRs 1 and 2. Do both PCRs is what we recommend now. Siravar specific bands, you can assign the Siravar. No Siravar specific bands, consider new serotypes. And that's when we get excited. It's not like stamp collecting. That's what people have said to us. This is stamp collecting. Well, it's not. It's clinically relevant worldwide. So we would disagree with you. And we got some strains from Germany. This was only a year or so ago. And these strains here were 9/11, or looked like they were 9/11, but some of them were very faint here. Very, very faint. And so we said, okay, let's try out a single-plex PCR. And we did that. And this is the single-plex PCR here. And you can see on the same samples with 9/11, you've now got a strong band. We have absolutely no clue why we are not getting a good PCR over here. We suspect The primers are not quite fit for purpose for some of these strains. We're obviously having those whole genome sequenced, and we are going to have to change again, I'm sure, to make it up to date and relevant to the industry. And that is what we will do. Ooh. Okay, so hopefully I've convinced you, formulated 2 PCRs, confirm and isolate as APP, detect all the recognized serovars, establish, confirm biovar status. And I also hope I've convinced you that this is worth doing. So we have made 0 money out of this, I'll be perfectly honest, none whatsoever. But there are plenty of people who are. So it's being used by companies worldwide. So here we've used examples, Anicon up here, they use it, IVD, they're both based in Germany. Or similarly, quantitative PCRs, Exapol in Spain, whole load of these. Lots of veterinary diagnostic universities as well, so I'm not sure what happens here in Viçosa, I've forgotten. So this is Iowa State. So actually, this does make a difference. It is used diagnostically. So we're pleased about that. We're not quite so pleased we haven't made any money, but, you know, hey, that would have been helpful. But I hope I've convinced you we've made a contribution. So what are the messages here? Genome sequencing, time and effort. We put a lot of effort into this. We struggle to keep up to date. I'm retiring soon. Somebody is going to have to take this on to keep it up to date because the companies won't. For sure, they rely on people like us in the universities. And that means for the students here, actually you can make a difference by doing things in universities that are translational, and the companies they will not modify this, they will just use them. And fine, that's good, it's good for us, but it does need, I think, academics to keep these things up to date, because they have the strains, I think, or prepared to do the strains. Will companies do it? I'm not sure they will, so we'll see. Anyway, there we are. Okay, so I'm just going to have a bit of water, and then we'll go on to part 2. Okay, so that was an example of a very simple diagnostic, PCR, yeah, that's made a difference. Now for something slightly more complicated. And this is a point-of-care test for the PRDC pathogens, the porcine respiratory disease complex pathogens. So I work in a pediatric department, so we have no veterinary departments at all in Imperial. There are just 1 or 2 of us who work in the veterinary arena. So I get exposed most of my life, I would say 98%, to human-related infections, etc. Yeah, I have to go outside for my veterinary hit. And the veterinary diagnostics, the human diagnostics are way ahead of the veterinary diagnostics, no question. And so for point of care, there are huge amounts of platforms out there, yeah, including for respiratory. Are they truly point of care? You can see here that actually there's some companies here. So, just trouble here. Abbott, Bosch, Quantum DX. So these are classed as point of care devices. Actually, this one's quite small, but they're mainly based in the laboratory. And so there are tests out there. So we're all now, I think COVID has actually been good for diagnostics, because I suspect every single person in this room has done a lateral flow test for COVID. Yeah, it's changed the mindset of people. So now you don't think about it. If you think you've got COVID or for some reason, you just go, I'll just do a test. Yeah. Whereas previously, lateral flow tests were, I would guess, the preserve of mainly women to see whether they were pregnant or not. Yeah, that was the one. So the whole has changed. So now people are comfortable with small tests. And there's a lot of these out there. A lot of big companies is in this space. And so let's just think about what you normally do with a sample. So let's just think APP, pig lungs. Here we go. Yeah. Normally you would send it off. Transportation, that'll take 1 to 2 days. In the UK, most of the lung samples, and I mean most of the lung samples, get sent to companies abroad, like Anicon, like IVD, because the turnaround time is much quicker. So 48 to 72 hours, much quicker than UK companies. That's bad. Yep. Then the sample might get stored a couple of days, DNA extraction, and then you can do your PCR and the results come back. So you're talking, if you're lucky, 48 hours, 72 hours plus to get a result. Yeah. So that's not great. So the starting point for this project that I'm going to talk about actually comes from a big grant that we had, this Brad Pitt consortium, which was to do with diagnostics and vaccines for 5 different bacterial pathogens. APP, that should say Haemophilus parasuis, now known as Glasarella parasuis, Mycoplasma pneumoniae, Streptococcus suis, and we had a strategy board who also recommended that we look at Pasteurella maltosida, and it will become clear why. So big consortium. So us at Imperial, University of Cambridge, London School of Hygiene and Tropical Medicine, Royal Vet College, Sanger Centre, that's in Cambridge, Animal and Plant Health Agency, so they're a government agency, DEFRA, government department in the UK, United States Department of Agriculture, and I've actually left off Zoetis here as well. They were involved as well. This was a big consortium, yeah? And so, let's go. And as part of that consortium, we were asked to come up with a multiplex PCR again for these 5 bacterial pathogens. So, again, so here's our pathogens up here, APP, Haemophilus parasuis, as it was called then, Mycoplasma pneumoniae, Strep suis. Actually, maltosida wasn't in our grant, but was recommended by our strategy board. So this was an international strategy board to include it. So we did, because they're our strategy board. And we wanted to identify species-specific genes from our whole genome sequence database. So we actually did over 720 genomes, about 200 or so for APP, 200 for HPS, 200 or 120 for MH, 200 for SS, and maltosida we had to get off the databases. And the strategy here for these isolates, again long-term here, for the UK isolates, we had ones from clinical cases associated with whatever the organism was, and we had ones from non-clinical cases. These came into the APHA. They did post-mortems, and they also swabbed the bronchi and the trachea. And if there was no lung involvement, clearly no lung involvement, but we isolated strains from the trachea or bronchi, we kept them back. So that was trying to answer a question. Are there strains for these pathogens which only colonize but do not cause disease? And this is about pathotyping. So PCRs to say, have you a virulent isolate? Have you a non-virulent isolate? So that was 2 years just to get those strains. Then the genome sequencing. So there we go. So then from all this data, big data, we heard about that yesterday, primers designed to generate 2 amplicons per species, and we also have to have an extra primer for divergent strep suis. So divergent, and we did come up with this PCR here, so 2 amplicons for each one, and here's a single amplicon for the divergent strep suis. What are divergent strep suis? So when we looked at the genomes, we could see most isolates were this normal strep suis, and we had a few which were virulent that were completely different. You can see from the profile. And it took us ages to get one single PCR to isolate these. Okay, so then we said, right, what should we do next? Yeah, we've got our PCR, what should we do next? So then we went to the industry. We went to the AHDB, Agriculture Horticulture Development Board. So this is a levy. So every time a pig is sold, there's a levy in the UK. And these guys, they do sheep, pork, cows as well. So they have a big stake in the industry. BQP, British Quality Pork, that is now owned by Pilgrims, and Pilgrims is now a Brazilian company. Is that right? I think that's correct. Outside of China, the biggest pig company in the world. BQP is the biggest pig company in the UK. Animal and Plant Health Agency, Susanna Williamson, so she is responsible for surveillance of UK pathogens in pigs. And CEVA, who had supported us, were happy to be involved. And they said, this was straightforward. They all said the same thing. What we need is a point-of-care test for the porcine respiratory disease complex because it will help us determine between viral infection and bacterial infection at the farm. Yeah. They also mentioned surveillance. I'm not so convinced by that. And this was the big driver. Big drivers in the UK to bring down the amount of antibiotics used, and this could help. So let's move on a bit quickly. So the porcine respiratory disease complex, it has our 5 bacterium, 3 of which are here, but it also has 3 viruses that are also big into this. So PRRS, porcine reproductive respiratory syndrome virus, swine influenza, porcine Zika virus. I have no experience in virology, so this was quite a big departure for us. And so that's what we're targeting. So 5 bacteria and 3 viruses. Can we come up with a point-of-care test you can do at the farm which targets all these? So a consortium was put together. So some of us at Imperial here, University of Cambridge, Dan Tucker, Paul and Eduardo at CEVA, Martin again at BQP, and Susanna, who is absolutely key to this because she can tell us what the up-to-date strains are, what we need to target. And 2 very important people here at Imperial are Pantelis and Jesus, and they were already developing point-of-care tests for human pathogens. There are so many platforms out there. Which ones to go for? I'd met these guys through another consortium to do with TB diagnosis in children. I liked what they were doing. This is serendipity, 2 coming together. So serendipity is about not only being in the right place at the right time, but the point for students is here, actually you've got to be able to make use of that time because it's, you come together, you don't talk, things don't happen. You have to make those connections, like the elevator pitch, essentially. Yeah, that's the message I would say for students. Okay, so what are these guys doing? Well, they were using LAMP, so this is another type of PCR, but the advantage of LAMP is 1, it's out of patent. 2, you can get enough, if you use a gel, in 30 minutes. You can do a PCR in actually less than 30 minutes. Yep. And so the difference with LAMP is, if you look at the NEB catalog, is that I'm not going to explain it. The difference is the temperature is constant, so at 60 to 65, you need 6 primers rather than 2. And what happens is you get these loops, and so with all these primers, and that allows this incredibly quick PCR reaction. Okay, so that sounds pretty straightforward. So what was the vision? Is to go from sample to pathogen amplification to detection in 30 minutes, yeah, at the farm. And so what they were developing at that time, and well developed now, was detecting the PCR based on pH, yeah? And that's because when you do a PCR reaction, you have your primers, and as the chains extend, you get protons released. Yeah? But these are incredibly small amounts of protons, really. So you need a detection system to pick that up. Yeah? And that's the clever bit of all this. And so they were working with these what are called CMSO chips, the chip platform. And CMSO chips have heaters and thermal sensors where you can just do lamp. It's easy to control the temperature. And the hydrogen ions that are produced can be electronically detected on this ion sensing field effect transistors. Now, I have tried looking at these papers. This electrical engineering is way above my head, for sure, way above it. But I do know it works. And this was their early device, actually. So here you have a chip, a circuit board. So this circuit board is made from components that you can just buy, actually, in electrical shops. So there's nothing fancy about this. And it is about the size of a phone, or was about the size of a phone. And the idea was, actually, once you have your result, you can just send it off to anywhere in the world. So that is to the vets in general. And you can monitor this reaction actually on the chips. So if you look at this one here, as green, you can see it on the phone. There's a program. You can see its positive result, voltage versus time. And if you have a negative result, you get nothing. And lots of clever algorithms convert this in real time, actually. So this works. And so the colors have gone here. So how would this work for us? Well, it was thought that one chip was actually going to be quite difficult, so we're going to use two chips. So we'll have a bacterial here and a viral, and then we'll do these LAMP assays for all the pathogens. And every single time these guys have done a LAMP assay in the lab, it's been convertible to the machine. Yeah, every single time. So it's easy. You do your LAMP assays, you do lots of different primer sets, you say, they're the best ones, let's move on. So that works. And so we now have LAMP assays, detection limits for all of these now. So all of our 5 pathogens, strep, suis, divergent, PERS 1 and 2, influenza, porcine circovirus. We've also got some for African swine fever. We haven't got African swine fever in the UK yet. It's only a matter of time. I don't think you have it in Brazil, is that right? It's not in Brazil. Okay, so there we are. And we have an internal control, which is the pig cytochrome B. And this one tells you that actually you can use not just pig lungs, but heart, joint tissue, which is relevant to strep suis, oral fluids, and liver. So this actually works quite nicely, this stuff. So we, like you, got hit very hard with COVID. We lost probably about a year with this project, I would say, with COVID, which was not great, but I think we're back on track. So let's have a look at some clinical data or clinical samples. So here is the number of genome copy numbers, various pathogens, and you can see the number of genome copies. This is from lung samples, is actually quite varied. Yeah, so that's good and bad, as you will see. But you can see there's quite a variation here in the copies. The quantification tells you that you can do quantitative LAMP. It's just as quantitative as qPCR. So here are some samples from the UK. This is just with the bacteria. There are multiple bacteria in a lot of these lung samples. This mirrors other people across the world. I suspect it's the same in Brazil. Actually, nobody's done this in the UK before, so there we go. Lots of the bacteria in these. Let's move on quite quickly. And there's a variable correlation between bacterial load and time to positive. So how quickly can you detect your signal? So this is APP, and you can see this is 2,000 plus genome copies, 200 to 2,000, 10 to 200. So less copies, longer time to detection. That looks like a line to me across there. Correlation. Here's Mycoplasma high and ammoniae, no correlation whatsoever. Yeah, so it varies. Time to positive. Okay, so We've done quite a few samples now, so it works quite nicely. I think we're happy with it. Apart from strep suis, these were the results that came out. A sensitivity of 45% is going to go nowhere. Yep, that is, everything else is comparable with other tests. So why is strep suis so difficult? I don't know, but there's absolutely a correlation. If you have more than 2,000 copies, then you have 100% sensitivity and specificity. So strep suis, you need to know. And here's a problem, which we found with all of these. Is there any data on load versus clinical symptoms, etc., in the literature? Answer, not anything in terms of genome copies, etc., with these guys. That makes it hard. I think that's the next study, yep, to try and correlate. Strep suis says you should be looking at this. The other one's an APP, possibly, but then we'd need all the metadata, clinical sample, clinical data, temperature, all that kind of stuff to be able to do that properly. We're not in that position. But I think that's the next study. Okay, so there's another way to detect this. You don't need to use chips necessarily. You can use a simple pH change. So we looked at 10 different dyes to do this. And the one that we've come up with a combination, which is quite nice. So this is APP. This is number of copies again. This is the control. So if it goes yellow, then that's positive. And if it still remains purple, then it's negative. And Jesus and Pantelis in March 2020, with the COVID, span out a company at Imperial called Proton DX here. And so they've now developed a system for this colorimetry test where you can do everything in 30 minutes. Yeah. So I haven't brought the kits here, but there's a very clever way of isolating the DNA. You do, you have a panel which has freeze-dried with all the reagents in and the primers. You have a little heater, which is very small, and you have a card to tell you. And this has actually been done with human pathogens, respiratory pathogens. It was done at the Beijing Olympics, which was in, can't remember. It's been a couple of years ago now. Used by the team GB, where it was for SARS, influenza, respiratory virus, human rhinovirus, and the athletes and the trainers were actually doing this. This particular one was a test that was done in a ski pod going up to the top of the ski runs. So not quite a farm. So the question for us, could we do exactly the same with the Dragonfly format? So first farm visit, which I missed because I had COVID. I'm still annoyed about that all these years later, but I am. And this was samples from rote washings and nasal swabs rather than lungs. And a kit was designed here. And this is Ollie. He's a PhD student who did this work. And you can see the heater is very small. It's actually designed, you can plug into your car. The newer version will actually have a battery, so you won't need anything at all. This is actually on a building on a farm, and you can see it's very small. And this does the amplifications. And when they went to the farm, they said, we think we've got swine influenza. Yep. Well, that was all negative, but what they did have was strep suis, Glasarella, and Maltosida in their lungs. So what would be the implication? What would you normally do? Here, they would have gone, we suspect influenza. We would send this lung off and say, for an influenza test. It would come back negative. Yeah. So cost-wise, a single test for SIV, actually with this format, you get the same for 8 pathogens for the same amount of money. And you don't have to send it away, and it's on the farm. Yep. So this format works, and we're happy with this. And so if you look at, this is just APP, we've done this all. This is quantitative PCR, quantitative lamp, colorimetric lamp. And you can see, if you just go down all these farms here, that you get correlation across here, except for this one, farm 9, where you have very low genome copy numbers. Yeah? But you can deal with that. That's up to the vets to deal with. So this works. So this kit is not out there commercially yet. And we're doing another test at the moment. This is with the format just with swine influenza, just the viruses, porcine circovirus and PRRS. And here what we're doing is we're taking a sample on the farm using Dragonfly, sending another sample off to the regular vets, that whoever the farm uses, what they're happy with, and we're also sending a sample back for genome copy number. And we should be able to put all these together. Yeah? So we have all the information. So that should be finished in 2 months' time. Okay, so that format, what's happening with the lacewing? So this is the chip version. Well, it's now moved on. So it looks like this. So the idea will be you have a single cartridge, you put your sample in here, you put it into the machine, it will do everything for you, and then it will send the results to whoever you want immediately with that. So the only thing that's holding us back at the moment is that for lung samples, you need something to mash up the lung samples. So there's a company in America who are designing exactly that, and that's not very far off now. So this, I think, will eventually, I think this will be a bit longer, a year or so. The Dragonfly format, that's easily doable. This one will take a little bit longer. Okay, so what are the messages here? Genome data, very useful, got to keep up to date. Engage with the end users. What do they want? Yeah, that's what's important here. What do they want? And so that's just can't, with this commercial stuff, that's what you've got to do. Work out your cost, etc. But there is a role for academics here. There is a role for students to do this stuff. Yeah, the companies may take this on and the rest of it, but actually you can work together. It's not always easy. You have different milestones, all that kind of stuff. But it's really worth doing. It's quite nice to see your research actually might make a difference, and that's what's happening. Okay, so last slide. Let's tell the people that have actually done the work. So Ollie, you saw in the picture with all the, he also was involved with the serotyping. Janine and Yamlin, Susanna, absolutely crucial with this. All these guys at Proton DX. Siva have been fantastic with us. Paul and Eduardo, Prevan, the bottom three here were the Portugal samples. These are a lot of the serotyping in the UK. Martin, who's now left, and money from the research councils, BBSRC, and Innovate to Proton DX. So I hope I've convinced you that all this was worth doing. So I am getting to the end of my career. It'd be nice if something got to the market. I think some of these are, but it is quite a nice feeling when you do something. And in my case, I've spent 30 odd years doing this stuff. Actually, there's something out there which I think is going to make a difference. Anyway, thank you for your time.
Thank you, Paul, for excellent presentation. So we will do the questions in the end of our lectures. Will be in the end of the day, okay? For now, we will do a stop for a break, a short break, and then we will return around 10 a.m., all right? So thank you, everybody, and see you in a few minutes. So, everybody, let's start again. And our second guest is the Professor Raúl Mainar. He's from Spain, from University of Saragossa, and he will talk a little bit of salmonella in the pig production chain. So, thank you, Raúl, and the stage is yours. Okay.
Well, good morning, everybody. I think first to say is thanks to the organizing committee for inviting me to be here. It's first time here in this place, so I'm enjoying very much. What I'm going to do is I'm going to give you some ideas about salmonella, the problem of the salmonella infection in the pig production. I will focus just on the pig production up to the slaughterhouse, no more than that. And, but what I'm going to present to you is some of our experience in Spain. Spain is one of the biggest pig producer countries in the world. And of course, each country has its own idiosyncrasy, you know. And we have our way of raising pigs. Every country has their way of raising pigs. And most of the studies we have developed has been in Spain. So what I'm going to show you is mostly based on what we have found in there, right? So I will start talking about, well, I will try to convince you why salmonella infection is important in pigs, despite that this producing nothing in the pig. I mean, it's mostly asymptomatic, and I will qualify that a little bit later. I will try to go through some ideas. I think they are important in order to understand why the difficulties of controlling pig salmonellosis in the pig chain, the pig production chain. I will go then to explain what some European countries have done so far trying to control the infection in the pig farms. And I will give you my opinion of why these programs didn't work. And of course, I don't want to leave this room without giving you some hopes on how to control salmonella, because I'm sure that you have the same problem that we have in Spain. So I will try to give you some potential ideas that you can work in your country as we have done in Spain. I have to tell you that I'm trying to convince you the importance of salmonella, but I couldn't get that from the Spanish people. They don't care about salmonella so far. The only people that care about salmonella are the slaughterhouses, but the farmers is not a priority so far. So let's start. Salmonellosis, I think, is very well known in humans. Everybody knows that when you get salmonella, you get a lot of problems, gastrointestinal problems. Most of the time, people have considered that salmonella is coming from the eggs or the poultry, contaminated eggs, poultry, and so on. And particularly if you use, how you call, mayonnaise, right? It's the same. We love that in Spain, and we make it at home with fresh eggs. And in the restaurants, used to be with fresh eggs. And when they were contaminated, then, well, the consequences were you get an outbreak of salmonella. So for many years, most of the things we have done to prevent salmonella in humans have been, well, try to prevent cross-contamination. I mean, try to prevent using or mixing raw materials, raw products with cooked products. And also try to cook very well the meat and the eggs and everything, so preventing the survival of salmonella. Well, this is more or less the human picture. I'm not a doctor, so I'm not going to talk more about that. But the picture in animals is quite different. We have been learning about salmonella in animals in the last, several years. But what we know is that salmonella can infect virtually any animal. Birds, mammals, reptiles. Actually, There are a lot of outbreaks of salmonella in US now with associated with reptiles, with turtles, little turtles that the people buy in the store and they play with them and they had salmonella. So it's a very common infection in animals. In some cases, some serotypes, I'm not going to talk about serotypes, I assume that everybody knows more or less what is a serotype, but it's just a different type of salmonella based on surface antigens. There are some serotypes that produce severe, important disease in animals. And let's see, we have some examples there. Abortus ovis will produce abortion in sheep. Abortus equi will produce abortion in equine. Cholera suis, Salmonella cholera suis is producing septicemia and very severe disease in swine. And there are some serotypes that can produce some severe diarrhea and gastrointestinal problems. But most of the serotypes are asymptomatic. The infection is invisible for farmers, especially or particularly in swine. And again, I'm talking about Spain. 95% of infections, of salmonella infection in pigs in Spain are asymptomatic. So we don't know they are there. And we don't have cholera suis, which is very common in the Americas. We don't have. There are some cases from time to time, but in Europe it's not very common. So we don't have a big problem with salmonella, where it's not a swine disease. And because it's not a swine disease, we have done nothing for many years. And when someone starts thinking about what happened with salmonella in pigs and doing something about that, the European Union decided, hey, let's check what is the situation of salmonella in Europe. And they did this baseline study, it was in 2007, in which they looked for salmonella in every member state. They designed the same study for all the countries because they want to see if they can compare the different prevalences in different countries. And they found that 10% of the pigs going to the slaughter were infected with salmonella. 10% infected means that they got lymph nodes, mesenteric lymph nodes, and they analyzed that, and they found salmonella in 10% of them. 1 out of 10 pigs going to the slaughterhouse were infected. Well, look at Spain. Spain had 30% of the pigs going to the slaughter infected with salmonella. I've been working with slaughterhouses that kill like 10,000 pigs per day. We have a slaughter close to my school that is, the capacity is for 30,000 pigs per day. There's no pigs enough for doing that, but they have this capacity. So 30% of the pigs infected with salmonella coming to the slaughterhouse. That means in average 10,000 pigs per day, 3,000 pigs per day are infected, are arriving to the slaughterhouse infected with salmonella. So, well, this was the situation for slaughter pigs. A year after that, they check what happened with the sow farms. They ran some surveys on multiplying sow farms, producing sow farms, any farms having sows. They analyzed fecal samples from the floor, from different facilities, from different pens, and they realized that almost 35% of the farms in Europe were infected, were contaminated with salmonella. Again, Spain and Netherlands in this case, we are having like a 60% of them. We repeat this study in 2011 in one of in our area in Spain, close to our school. And I can assure you that 100% of the fattening farms are infected with salmonella in high or low levels, but you will find salmonella in all of them, in all the fattening farms. Well, so it seems that salmonella is in the slaughter pigs, it's in sow farms, and what until a few years ago, we didn't know what happened in other phases of the pig production chain, and that is maternity. What happened with piglets? Because the few papers talking about piglets usually are experimental infection on piglets, and they say that the prevalence in piglets is very low, mostly because of these maternal antibodies that the pig will get from the sow will protect the pig at least for 8 weeks or so, between 6 and 8 weeks. Well, you know that in Spain we love piglets. I mean, loving in the sense of we eat piglets. Roasted piglet is a wonderful thing to taste. And because of that, we have slaughters devoted to slaughtering piglets. So we have the opportunity to go to one of these slaughter facilities and get the gastrointestinal tract of, I think was like a 900 piglets, 4 weeks old. We had the possibility of getting as much of fecal content as possible. We got all these small lymph nodes, mesenteric lymph nodes that they are, these like lentils. We spent a lot of time just trying to pick up all of them. So we have a very nice sample from each piglet, and we found that on average, 36% of the piglets were infected and shedding salmonella. And they were okay. I mean, they went to the slaughterhouse. They were not treated with antibiotics because they were to the slaughterhouse. They went to the slaughterhouse. So they are perfectly healthy animals, even being so young, even without having a good immune system, they were healthy. So I'm sure the salmonella is spread throughout the whole pig production chain. And the question is that important? I mean, because nobody cares. Then we have to go to the other part of the story, that is the human cases of salmonella. And if you look at the statistics from Europe, from the European Union, salmonella is the second most important foodborne zoonosis transmitted through contaminated food. And actually it's the first one when we are talking about outbreaks. Seems that when salmonella hit a lot of people at the same time. It's not like Campylobacter that is infecting one people sometimes, or another. So salmonella is an important pathogen in Europe. When we look who is producing salmonella in humans, the major serotype, the most important serotype is Enteritidis, and the second one is Typhimurium, monophasic, and then we have some others. And actually, many others are producing some infections, but they are not so important. But basically, one of the messages to take from here is that any serotype is potentially pathogenic for humans, right? So, the evolution of salmonellosis in human cases of salmonella in Europe has been something like that. From 2005 to basically 2013, more or less, there was a big significant drop in cases. Then there was some sort of stabilization of the number of cases. Well, this is the COVID thing and the Brexit. We are not counting now cases in United Kingdom, so there was this drop here. And now, after the COVID, seems that we are recovering some cases from salmonella cases, right? But if you look at this and this, you see that these lines are parallel. And this is because the reduction of cases of salmonella in humans is mostly because of the national control programs put in place in European countries against salmonella in fowl, in birds, trying to prevent the contamination of eggs and poultry. They have been very successful. The prevalence of salmonella in birds right now in Europe, in most of the countries, is very low. So we got this decrease in salmonella enteritidis, which is coming mostly from from birds. But look at Typhimurium, it's kind of more, well, there is some drop because for these programs we use vaccines that are targeting Salmonella enteritidis, but also Salmonella typhimurium in birds. So we can find some decrease in Salmonella typhimurium here. We have from 2007-08, we start seeing some new cases of salmonellosis coming from a new serotype, which is called the monophasic variant of typhimurium. And this has been growing up, steadily growing up. And this is because, well, there are some theories about that, but some have some advantages, this serotype. One of them is that it's multi-resistant. So this is the picture of the evolution of the Salmonella in humans. In 2005, they started with these national control programs in birds, and we decreased that. So actually, the European Union was thinking about, hey, if we do something similar in pigs, if we start some national control programs in pigs, maybe we can just get this down. Okay. So that's the reason of previous surveys I showed you. So looking at what serotypes are affecting pigs, I'm showing you some studies from Spain. This one is one of the last we have from our team. Typhimurium has been, for many years, the major serotype, but now is the monophasic typhimurium is getting attention. It's getting more and more infections in pigs. In European Union was something similar, but this is kind of outdated data. But we are seeing that trend. The monophasic variant is increasing. Typhimurium is more or less remaining there. Enteritidis is coming down, although there has been some increases in the last years, maybe some failures on national control programs. When we look at the origin of these serotypes in human infections, enteritidis is coming mostly from birds, broilers or layers or turkeys. Okay, there is no point about that. There is some enteritidis in pigs, but it's just a few of them. Well, infantis is coming, is the fourth or fifth serotype, is coming also mostly from broilers. But when we look at typhimurium and monophasic typhimurium is coming mostly from pigs. And typhimurium is shared by broiler and by pigs, mostly. So this is the situation. So the pigs have an important role on transmission, or this is what we think. And actually there was some modeling study a few years ago that they determined that 43% of the cases of human cases of Salmonella should be attributed to birds, to layers, to eggs or poultry, and almost 27 will be attributed to pigs. That in the whole Europe, on average. But when you look at southern countries like Spain, pigs may be behind almost 44% of the cases, of human cases. And I'm talking about Spain, but maybe, I don't know, Claudio can tell us, but maybe Italy, Greece are in the same pattern. So, what do we expect from this? And, well, we don't have to expect anything because it's already here. I mean, the first thing that happened is that there were some countries that some time ago, they started to think about Salmonella in general in all domestic animals and started to do things. And they got almost free from Salmonella. They have very good health status regarding Salmonella and some other pathogens. So these countries are Finland and Sweden, so they got from the European Union some advantages. European Union recognized that they were ahead of other countries in health status. So there is a regulation that allows them to deny the entry of meat coming from countries that they consider that they are not having the same standards that they have. So there is a real sanitary barrier there for these two countries. I know that the Asian countries are putting many problems to the importation of food coming from third countries like Spain. Even they don't have the same high standards, but because they are out of the European Union, they can ask what they want, right? Another thing that is happening, has happened and has stopped to happen, is that some European countries, I will talk about this later, but some European countries started to think about doing something, and they initiate some national control programs against Salmonella, against pig salmonellosis. And when they started that, all the farmers within the program got this quality label that allowed them to, well, get better prices, I would say, for their products, or being able to export more easily than those farmers that were not within the program. So there is starting to have differences regarding if you are or not within. It's not working? Oh, it's red, yeah. Okay, better? So there are quality labels that are going to tell you you are a good farmer, you are going to sell your pigs at a good price. You are not a good farmer, you are going to get penalized in some way. I think that Brazil is in the same situation as us. We are the third country, the third major pig producer country in Europe, in the world. But Brazil is the fourth. And I'm sure you have to export pork to other countries. So you may have the same problems that we may have in the future. We are not having so far these problems, but I think it's something that we have to think about. You have the same problem we have regarding Salmonella. Well, you have more problems than us, because you have Salmonella cholera Suis. We don't have cholera Suis, so you have a public health problem and you have a swine problem, both. You have to fight against both of them. But again, monophasic typhimurion and typhimurion are the most prevalent serotypes in pig production in your country. And I got this article is regarding antimicrobial resistance, but I love the title. I think it's the same for Salmonella. Are we wait to the market to tell us what to do, or we should be ahead of the market and try to do things before they ask us to do them, right? I mean, are we waiting for markets or not? If we wait for market, there will be some time in which we will not be able to sell pork. And this is the question I try to explain to people in Spain, but they are not, now I have to be very close to this. Wow, I'm eating this microphone. Okay, so hopefully I convince you about the importance of Salmonella, right? Let's go to the epidemiology. It's going to be very brief. I just want to point out some important things. I mean, I assume that everybody knows more or less Salmonella. It's a gram-negative bacteria. It's very ubiquitous. It's in all kind of animals. It's in the intestinal tract. Most of them, they will be shed by feces. The feces will contaminate the environment, and the environment, in the environment they will resist. It's very resistant. It will have an ample range of temperatures in which it can survive and grow. Or it has also an ample range of pH. And, well, Spain has a good climate for Salmonella outside. But Salmonella love this country, I guess. I mean, humidity, warm, everything is good for Salmonella outside. So Salmonella is in any place. You can find Salmonella in any place. So when you think about a pig farm, the first thing you have to think is, how can I get Salmonella in my farm? And there are many sources of that. Valentina will talk about one of them, the feed, contamination of feed, right? But there are many others. Wild animals, rodents, wild birds. We did a study on wild birds, and they got the same Salmonella that was in the farms. Actually, they were the victims, not the one bringing Salmonella to the farms. Insects can mechanically transmit Salmonella. Trucks entering and exiting the farm, new replacement, the people just working in the farm, entering and exiting the farm. If there are another farm closed and they don't change food, they don't change clothes. I mean, a lot of things. And this is what we call external biosecurity, right? And it's important to try to keep those factors down, but this requires an effort by the farmer, an effort that usually is related to the way they work, the routine work they do. And they don't care about that, because how you convince someone to do something that is good for something that they don't see? I mean, when you have a disease and someone tells you, do this and the disease will be gone, you do, and you see the result. But here, you will not see anything, right? Then once it's in the farm, Salmonella is very easily spread between animals. But again, there will be some factors, internal factors, that will help to this dissemination. And some of them will be the same as before, and some others will be related with how you handle the animals. Stress is an important factor for Salmonella shedding. One animal will get infected, will shed some Salmonella at the beginning, but they may stop shedding. And they may never shed again unless it gets stressed in some way. So stress within the farm will help the shedding of Salmonella. Like other diseases, particularly those gastrointestinal diseases, will be affecting the shedding of Salmonella and the transmission of Salmonella in the farm. Okay, and at the end of that, well, this is within what we call internal biosecurity, all the preventive measures to try to prevent the transmission within the farm. And then finally we have those pigs that are in the farm, some of them are infected, and we will take them to a truck. We will drive them for, I don't know, a couple of hours. Here, the roads are difficult. I mean, for a truck, you spend a lot of time on the road to reach the slaughterhouse. During that time, the pigs are not familiar with driving, you know, so they are kind of stressed. They reach the slaughterhouse, they will wait there for some time, and they will again be stressed. And those animals that are infected, it's very easy that they will start shedding Salmonella. And remember, in Spain, 30% of them are infected. Okay? So this is the picture of the epidemiology, the simple picture. Another important point is the diagnostic techniques. We have been talking about PCR in the previous class. Let's see what happened with Salmonella. In Salmonella, we have a reference test, which is bacteriology. There is many different protocols for isolation of Salmonella, but in Europe, we use one. The one here is the standard protocol that we use because I guess it's comparable among countries, right? It's a test that takes some pre-enrichment, selective enrichment, some isolation, and some biochemical confirmation. It takes minimum of 4 days, okay? So it's not quite easy. Actually, it's very slow. You require fresh samples. You require fecal samples taken within the last 24 hours. It's kind of expensive because it's taking time. You need people doing this. And the sensitivity of the test is limited, particularly when we are talking about asymptomatic animals, okay? Because remember, not all of them will be shedding, okay? And even when they are shedding, some Salmonella will be weak. There will be low loads of Salmonella in the feces, so it's not as simple. When we work on lymph nodes, which is a more clean sample, sensitivity is quite good. But when we work on fecal samples, there are a lot of things going on there, a lot of bacteria, and small amounts of Salmonella. So the sensitivity usually goes as low as 50-something, and sometimes it's 90. Depends on the size of the sample. I mean, this technique, this protocol requires 25 grams. Less than that is limiting your sensitivity. The advantages of this is that it's 100 specific. If you find Salmonella, you did nothing wrong in the lab. This Salmonella belongs to this animal, so that's good. And you can serotype. And serotyping is important because, as you saw before, there are some serotypes that are more important than others. Okay, serology is the other technique we use. It's much quick, it's cheap. We use small volumes of sample, usually blood, well, serum, but also meat juice. You can freeze the sample and analyze when it's more convenient for you. You can include several antigens in pigs. So in theory, you will detect any Salmonella affecting pigs. And there are several kits already commercial that you can buy, and it's very simple to run them, right? Of course, there are problems. When we did some comparison between ELISAs a few years ago, we found that they say different things for the same sample. So, wow, we have to, I don't know, we need to correlate in some way these different tests. We already did some tests comparing what happened when you use serum instead of meat juice, or comparing meat juice and serum. And we found that the values, the ELISA values for meat juice were usually lower than for serum, which makes some sense. So basically, what I'm telling you is, if you are going to use ELISA, you need to make sure that you are using similar ELISAs, and you are using the same type of, same matrix, the same sample. Because if you are changing samples, you may have some differences, right? But the problem with ELISA is that there is no correlation with shedding, even with infection. There are many studies trying to determine the correlation between infection in a pig and serology, and the correlation usually is low. In part because, as I said before, pigs are not shedding salmonella every day. But also because there is a lack of sensitivity and specificity of the test. If you look at this table, I don't know if you can see from there, but the sensitivity and specificity is quite low, around 70 or something like that. So there is ample room for mistakes. So it's difficult to interpret individual analysis, serological analysis. In theory, or in general, we use serology when we are working in batch of pigs, in many pigs. So if we have a lot of them with high values for the ELISA, probably we know that salmonella is circulating around, okay? And then we have the PCR. I'm not going to talk much about PCR because PCR has the same problem as bacteriology. We need to enrich the sample if it's from an asymptomatic. If we are talking about symptomatic animals, it's fine. I mean, bacteriology will work, serology probably will work, and PCR will work. But we are not talking about symptomatic animals. We are talking about asymptomatic animals. So we'll have to do some enrichment. It will take 24 hours, and then we may run the PCR. But getting the PCR from this buffer peptone water is kind of complicated. I don't know why, but there is something going on there that is maybe many inhibitors or things like that that make the PCR is not working well. It's working very well in food, and it's working very well in lymph nodes because it's just meat. I mean, it's a very clean sample, but it's not working very well in fecal samples. So I guess sensitivity and specificity in lymph nodes is okay, but I guess in fecal samples we have to work more on this, right? The other things, well, you can see is quick, is more or less the cost of the bacteriology. Well, so basically these are the three tools we have so far. And I want you to remember the bacteriology is good, but we'll underestimate your real prevalence of infection. Serology is good, but if you work on pig with large number of pigs, just to have an idea of circulation, you have to be very careful with the kit you use. And the end of that is that if you really want to know what is going on in a farm, you will have to use both serology on a sample of pigs just to know if salmonella is circulating, and if you consider, based on your results, the salmonella is there, then you will go to get samples, fecal samples from the pens, floor, whatever, and try to identify what serotype is in there. Because if it's one of the biggest, is it typhimurium, monophasic typhimurium enteritidis, then you have a problem that has to solve. If there is one exotic serotype, maybe it's not as important because many of them are coming and they leave without notice. I mean, it's not, they don't remain in the farm. But a typhimurium serotype will remain for years if you don't do anything. So now the next question is, okay, what happened with the abattoir? What happened with, because this is a common discussion in Spain. Producers are blaming the slaughterhouses about what they are doing wrong to get so many salmonella in the carcasses, And slaughterhouses are blaming the producers because they are bringing pigs with salmonella. And they are always discussing that, or arguing about that. So let me put some clear things on this. Okay, the problem in the slaughterhouse and the problem for the people is having carcasses that are contaminated with salmonella. Where this contamination is coming from? Of course, from the pig that is infected. An infected pig, we have salmonella inside, lymph nodes, tonsils, gastrointestinal tract, but will have or may have salmonella outside because it's contaminated itself with fecal material, right? Usually, slaughterhouses usually do many things, and I think at least the most modern slaughterhouses are doing things very well. They wash the animal after killing it, they scalding them. If you keep temperatures high, 63, 64, 65, in the scalding, the water for scalding, it will be good to kill most of the bacterial contamination outside. Thinking to burn the hairs and also burning any piece of bacteria that is there. Evisceration is an important point. Mostly the cut is done by an automatic machine, and usually it's very good. And then there will be some operator just dragging the gastrointestinal from there. Sometimes there's ruptures. If there is a rupture of the intestine, there will be some fecal material going out and probably contaminating the. So this carcass has to be out of the line. And then there is another thing that we stopped doing in Spain is the split of the head, because if you cut the head by the middle, you can touch the tonsils and you get some salmonella contamination in the plates that can go through other carcasses. So now we just put the whole head to one side. Okay? And the other source of contamination for the carcasses is other infected pigs. Prior to this one, that were infected and were shedding, or there was some environmental contamination, and for some reason did arrive to the carcass. Of course, people going in and out from the dirty zone to the clean zone. Rodents, if there are rodents in the slaughterhouse, usually they are not. I don't know, some lack of hygiene, lack of disinfection at the end of the day. Things like that are the ones that may affect the carcass contamination. But in general, I think they do very well job. The slaughterhouses usually, they do very well job. So basically, the major source of attribution for carcass contamination is a pig shedding salmonella when arrived at the slaughterhouse, because it's going to be the origin of the contamination in the slaughterhouse, right? Okay. We did some studies trying to identify what was the relationship between the health status of the animal in the farm and the shedding at the slaughterhouse. And we found something interesting. We follow several batches of pigs from one farm during the whole fattening, growing, finishing period. This is 3 months, more or less. We were taking serum samples. And at the end, when they arrived to the slaughterhouse, we got some fecal content to see if they were shedding, and also lymph nodes. And we saw that when pigs were always seronegative, and there was no salmonella in the lymph nodes, the background shedding was more or less 20-something. So 20-something pigs, 20%, 25% of the pigs may shed salmonella if they were seronegative and there was no lymph node positive in that pigs. Again, this may come from, or this apparently high level of shedding for those that apparently are negative pigs may come from the lack of sensitivity and specificity of the test we use. So we may not find salmonella, but it's still there, or these animals were infected just when they arrived to the slaughterhouse, something like that. Okay? But the most interesting thing was that when we had pigs that were seronegative during the whole period, but were seropositive at lymph nodes, the odds of shedding was almost 5 times this. And if they were positive at least once during that period of growing, and they have seropositive, positive lymph nodes, the odds of shedding was 21 times higher. So there is a clear relationship between the health status of the animals in the farm and the probability of shedding Salmonella when they arrived. Actually, we found that those shedders seroconvert much before and remain with high OD values during longer than those animals that were not shedders at the slaughterhouse. So it seems that there is some relationship between the health status in the farm and the shedding. So something has to be done, I guess, in order to prevent the contamination of the slaughterhouse. I mean, we have to help the slaughterhouse, and the slaughterhouse has to do their work on the slaughterhouse. So, I mean, both have to work together in some way. Okay. So next is, well, what other countries have done so far? I told you that Spain has done nothing so far, right? But there are some countries that start doing things. Okay. There was particularly those Scandinavian countries that, because some outbreaks of Salmonella in people some years ago, they decided that they want to get rid of Salmonella from domestic animals. And they start a very, very strong campaign against Salmonella. And they basically are free of Salmonella right now. They just depopulate herds with Salmonella, they do serology, they do bacteriology, they do anything they have to do just to get rid of animals that were infected with Salmonella. But hey, they are Scandinavian. I mean, climate is in 6 months of cold, few pigs, few people, few farms, few everything. It's easy to work on that, right? Now come to Brazil and do the same. Start depopulating farms here or in Spain. It's impossible, right? Denmark did something similar. They start pushing for eradicating Salmonella, and they start working very hard. They spend a lot of money doing that, and they got some sort of success. They have very low levels of Salmonella right now, but I heard one of these guys behind the program saying a few months ago in a conference that he will not repeat again because they spend so much money and they still have Salmonella. So there must be some other ways to do this. And then because Denmark was pushing to the European Union to, well, try to get things better and lower prevalence of Salmonella, some other countries follow suit. Okay, United Kingdom, Ireland, Germany, Netherlands, Belgium. I have to tell you that, well, there was some success for these Scandinavian countries. Germany, a couple of years ago, I think, they show for first time some reduction in the number of farms positive to Salmonella, but after 20 years working on the program. So I don't think has been very success. We don't have news about Ireland and Netherlands. I think, well, there is no news, means they don't get anything, because if you get something good, you will publish it. And there were some reports, and United Kingdom and Belgium just stopped the programs because they didn't get anything, right? So this is the situation. What was wrong with this? Well, these countries, instead of going for eradication, as Denmark tried and as the Scandinavian countries, they decided they will go for control, meaning that they will try to reduce the prevalence of infection, but not to eradicate the Salmonella, because we know that it's basically impossible right now. So what they did, they worked mostly with growing, finishing pigs. They used serology, and they got some samples, a few samples from each farm along the year to determine the levels of seroprevalence. Basically, what they did was they got these repeated samples. They get some average seroprevalence, weighted average, because it's giving more weight to the last sampling. And according to the results of this average, they say, okay, this is a high-risk farm, high levels of seroprevalence. This is a low-risk farm, or this is a farm in between, right? And they start telling the farmers from high-risk farms to do something. I mean, they start checking biosecurity, facilities, everything, and telling the farmer, okay, you have to change this, you have to change this routine, you have to do this thing in this way, you blah, 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 blah. A lot of things that the farmer has to do and keep forever, basically, because Salmonella is out, it's there. I mean, you have to keep those things going on. In my opinion, these programs failed for a couple of things, well, many things, but three major things. First, they took a small number of animals to determine the seroprevalence of large herds. I mean, everybody knows the importance of sample size in order to determine with some precision the prevalence of something, right? And they were taking 60, maybe 70-something pigs along the year to determine the prevalence of a farm. In Spain, a farm may send to slaughter maybe 4,000 pigs in 1 year, depending on an average farm of 2,000 pigs, but at least 2,000. 60 pigs to get the precision for 2,000, I don't know if it's very good. But in addition to that, in many cases, they got the pigs at the slaughterhouse. The distribution of the pigs, when they arrive to the slaughterhouse, is not the distribution of the pigs in the farm. So they were not representative of the unit in the farm. That, I think, is another problem. And then remember the diagnostic accuracy of serology. Sensitivity and specificity in general is low. So I think there are a lot of problems. In addition to some others, like in some countries, the program was voluntary, in some others it was compulsory. Some characteristics of each country, I mean, some idiosyncrasy from the farmers. I mean, there are a lot of things going on there. But the thing is that instead of having a good precision of what's going on in the farm, I think what we are getting from these programs is a misperception of the real health status of the herd. And then nobody understands anything. Because when the farmers are trying to do something and they get nothing, and they get nothing because they get positive samples, they get very frustrated. And ask this guy to, okay, you go ahead, continue with your new routine, spend some time on this, that you at the end you will end up. And they are not getting this, and at the same time, they don't see any difference in the farm because the animals are okay. So it's very complicated to transmit the importance of this. So just to finish then, what to do? It's quite complicated. So we were thinking, because Spain sometimes it was deciding, okay, let's copy the program from Germany, United Kingdom, say, no, don't do that, please. But they try. I mean, there was some region in Spain that tried to do the same program. They failed. They last for 6 months because nobody was interested in that program. Something different has to be done. And when we are thinking about that, we asked for a proposal, project proposal to get some funds to study something. And we want to change the perspective of the control programs. We know that Salmonella is not a pig problem, it's a public health problem, right? So maybe we have to look in another way. We also know there was some studies, previous studies that show that the amount of Salmonella in the intestines was related to carcass contamination. So basically what this is telling us is that the pigs shedding Salmonella at the slaughterhouse are the main responsibles of the infection in the carcasses, the contamination of the carcasses. So why not try to reduce shedding at the slaughterhouse, no matter what happened in the farm? Let's try to stop shedding when they arrive to the slaughterhouse. So for that, we wanted to do something. We wanted to see if we can predict if a pig is going to shed Salmonella when it arrives to the slaughterhouse. And we wanted to do that because in a previous study, we saw that there was some relationship between serology and shedding at the slaughterhouse. And I told you before that there is no correlation between serology and shedding, but there's no correlation when you take the sample at the same time. This pig, I got the blood sample and I got the fecal sample, there is no correlation. But if you get, if you get your sample, your blood sample at one point in the farm, and then your fecal sample in the slaughterhouse, in the middle there is a period of stress. And if the animal were infected before, it's very likely there will be shedding. So we found this relationship between serology, increasing serology, and increasing the probability of shedding. So we want test this, but in a large scale study. So we went to 30 unit farms or unit fattening units in the northeast of Spain, which is one of the large pig production areas in Spain. We went to the farm. We did this questionnaire about biosecurity. The veterinarian in the farm was that for us. The survey was based on Biocheck, University of Ghent. It has an online questionnaire for biosecurity, very interesting one. So we did that for each farm. We took samples from 50 pigs, serum samples from 50 pigs, and we took 10 samples from 10 pens in the farm, evenly distributed in the unit, all the pigs and the pens. Okay, we tagged these pigs, and when they arrived to the slaughterhouse, we were waiting for them. And we got fecal samples from these animals, and we analyzed if Salmonella was there, if they were shedding. We checked if the strains found here, Salmonella positive samples here, if the strains were the same as here. Mostly Typhimurium, the monophasic, we were interested in. We checked that for PFGG analysis just to see if genetically they were the same. Then we ran a logistic regression model to predict if shedding was related to serology, individual serology, farm biosecurity, the positive Salmonella positive pens, the number or the proportion that were there, and some other variable like distance to abattoir, the presence or not, the Typhimurium, season, something like that, right? So we checked the biosecurity of our farms and was more or less okay. It was around 73%. It's just a score. As close as you are to 100%, better is your biosecurity, right? It was good. I mean, there was some farms with low biosecurity, but we can blame the biosecurity for the results we got, because actually, when we check what was the average biosecurity for many farms in different countries, we were above many of them. So they were really good farms in general. We detect in 43% of the farms, we detect Salmonella, and in half of them, there was more than 3 pens, or more than 30% of the pens positive to Salmonella, right? Typhimurium and the monophasic were the most important serotypes in the farms. We checked for serology, and we found that in most of the farms, the average seroprevalence was 30-something, 31, 32%, and just a few of them had very low seroprevalence. So that means that the Salmonella was circulating in those farms. So, okay, we have Salmonella in the farms. Let's see what happened in the slaughterhouse. We found that 24 of the pigs were shedding Salmonella, 24. I said before that we have 30% of the pigs infected with Salmonella. Well, in this study, 24 of them were shedding Salmonella. Makes sense, more or less. Most of them were Typhimurium and also monophasic Typhimurium and Typhimurium, so were the same strains that we found in the farm. And actually, when we check in those farms that we found the same strain, we check if they were actually the same with this analysis, and in 80% of the farms, we found a match. That means that what is in the farm ends up in the slaughterhouse, okay? So we went to the model. We want to see what the model was to tell us. We use, almost 1,400 pigs, but 2/3 of them were used for the model, and the other 1/3 was used for a validation model. I'm showing just the model, not the validation one. And we found a relationship with serology, with the percentage of Salmonella positive pens in the farm, and also with the biosecurity. As the biosecurity increased, the odds of shedding Salmonella decrease, which makes sense. As percentage of Salmonella positive pens increase, shedding in the slaughterhouse increased a lot. And also there was some positive correlation between serology and shedding Salmonella. So these 3 factors predict in some way the shedding of Salmonella in the slaughterhouse. Actually, when the model gives us, for a pig, a probability of shedding of 26 or more, we consider this pig positive. If the probability of shedding for that pig is less than 26, we consider the pig a non-shedder. And with this cutoff, we have a sensitivity and a specificity of 70-something, 72, 74, which is, well, it's not bad, considering what we got with other tests. So we have an opportunity of determining if a batch of pigs will, or important proportion, we have to decide what we consider now a batch of pigs shedding Salmonella slaughterhouse. Is 10% of them shedding Salmonella is big, or 20, or 30, or 40? That has to be decided, right? But we have, with this model, we have some opportunity to determine that. Actually, we ran the model with the different scenarios that were possible. And what is interesting to see is that when the farm has very good biosecurity, when I'm talking about biosecurity, it was internal biosecurity. External biosecurity didn't correlate with shedding, but internal did. And when the pigs had, or the farms had high or good biosecurity, internal biosecurity, and there was no positive pens in the farm, actually, serology is not needed, but because all the pigs are going to be below the threshold. So farms with high biosecurity and with no Salmonella in the pens, 30, I forgot to say that we took the samples at the farm 1 month, 3-4 weeks before going to the slaughterhouse. So we have 1 month ahead. So if 1 month ahead we don't find Salmonella in the farm and the biosecurity is okay, we don't care about these pigs. They are okay. But if the pigs are coming from farms with low biosecurity, internal biosecurity, and there are some Salmonella in the farms, we don't care about serology because all of them are going to be far away from the threshold. We have to consider them as potential shedders when they arrive to the slaughterhouse. Okay? And then there are some other scenarios in which serology will give us more details about the probability of a pig having or not, or shedding or not Salmonella. So it's a model that can be used in some way. And the advantage of this is that you don't rely on farmers. I mean, you go to the farm, you get your samples, you get your survey on biosecurity. You don't need to rely on the farm or what the farm is, the farm's opinion. I mean, this is kind of objective information that you can apply for the model. So what next? Let's say that we find a batch of pigs that are having Salmonella or are going to shed Salmonella, or we decide that they are going to shed Salmonella. Well, we define this as a high-risk batch. If we tell the slaughterhouse, hey, there is a high-risk batch coming to the slaughterhouse, okay, they will do some logistic slaughter. They can keep those animals to be slaughtered at the end of the day or at the end of the week. They can put in another pen at the slaughterhouse for some time. They can stream the hygienic measures. They can make sure that the truck that is transporting these animals will be very well disinfected and clean. And anything else? Well, we checked something else that could be work. Was what about if we put these animals in a pen in the slaughterhouse with water treated with some organic acids? Will that reduce the shedding for those pigs? So we did this study. We looked for we tried several organic acids. Some gave some taste, some like a vinegar and things like that. They don't taste well, and I don't think the pigs will drink much water while they are at the light age. But we found this esterified formic acid that can be diluted in water. They don't taste, have no odor. They don't taste. It doesn't taste. It doesn't change the pH of the water, and has some advantages. I'm not chemistry. I don't know anything about chemistry, but it seems it has some advantages because it works around all the intestinal tract. So say, well, let's try a high dose of this in the water when they are waiting at the slaughterhouse. And we are going to keep them overnight. They are going to be there at least for 10, 12 hours. And we found that there was some significant reductions, and overall there was an important reduction on shedding. And that was just for a few hours. I mean, wow, well, maybe something that we can think about. But what about from the point of view of the farmer? What to do? The same. If we determine 1 month before going to the slaughter that the pigs are going to be shedders, we can start doing something at the farm. And most of the treatments have been thought to be done on feed. But in this case, you know that the pigs are going to go through fasting before going to the slaughterhouse. So if you put the treatment on the feed, there will be some time they will not eat, and there will not any effect from the treatment. So we thought, much better in water. If we put in water, they will drink water until the end, okay? So we used the same organic acid because it was kind of good in the slaughterhouse for just a few hours. What happened if we put for some days before going to the slaughterhouse? So we put the same organic acid, the same dose for 5 days, and we found very significant reductions. Actually, we found an efficacy of 82% in the reduction of Salmonella shedding in the group that was treated. So imagine now that we combine this with this. We may get a significant reduction of Salmonella, okay? So just finishing, we think that we get an acceptable knowledge on the probability of shedding with this model, and this information, of course, can be very useful for the farmer and for the slaughterhouse. And we can start working here. We have some advantage. If we are doing this for any batch of pigs going to the slaughterhouse, at the same time that we know what is the situation of this batch, we know what is the situation of the farm. So we can classify the farms as well as before, but having representative samples of the farm, okay? So I think we can get some good ideas from here. And just to finish, I would like to thank, well, of course, big companies, slaughterhouses, veterinary students have been working with us in the slaughterhouse, just taking samples. My PhD students along these years, I just put one of the funding projects, but there has been several working on this. And that's all. I mean, questions for later, I guess.
So thank you, Raul. The questions will be in the end of our lectures, okay? So now I would like to invite Professor Valentina Trineta from the Kansas State University and for the next lecture. So welcome to stage.
Yeah, thank you. So Raul, I just wanted to tell you that you convinced me. So while Raul told you something about the Spanish experience, I'm gonna tell you a little bit about the situation in the United States. My name is Valentina Trinetta, and I'm originally from Italy, but I am working at Kansas State University. My presentation has some overlapping with Raul, but my perspective is more from the feed production point. So, a couple of questions for you. Actually, one question, because when I got prepared for this presentation, do you know #1 foodborne pathogen in Brazil that cause outbreak? That's the title of my presentation. That's the title of Raul's presentation. So, Salmonella. So Salmonella, almost 23%. And I don't know, maybe Nero and Luciano have more updated data. This is something that I found published in 2020, but was until 2018. You guys have a lot of problem with Salmonella. You have problem with a parasite, trypanosoma. You have problem with norovirus. From what I understood, water is huge, red meat, poultry. A lot of contamination happen at home, restaurant. There are not a lot of data on recall. That's something instead that in the United States we are pretty big about. So a lot of similarity with United States. Number 1, foodborne pathogen, norovirus. Number 2, Salmonella. If I look at the economic impact of this foodborne pathogen, Salmonella is much higher. We spend a lot of money in the United States to recover from Salmonella and Listeria as compared to norovirus. So that's why Raul already convinced all the Americans because we know that we spend a lot of time, a lot of money for Salmonella. If I need to look where I get Salmonella, poultry, eggs, pork, and beef. There is a huge restructuration of legislation of Salmonella in United States for poultry. I participated to the committee that was putting together the legislation, and we tried to imitate Sweden and the Scandinavian country, but where we produce poultry in the United States is mainly on the south, so it's not going to work out. So we are trying to work a lot on vaccine. We are trying to understand what are the point of the chain that increase Salmonella. We are trying to understand what we can do from feed to fork. Okay, so just a refresh for you guys students, okay? Gram-negative, facultative anaerobe, likes a lot 35, 37 Celsius, that means it's our body temperature. So we are the perfect carrier for Salmonella. I want to review this one because my study will talk a lot about Salmonella monophasic. And I will tell you a little story about this bacteria that apparently migrate from Europe to United States. But if I start to swab and collect environmental sample here, I'm gonna increase my personal collection of Salmonella because Salmonella is everywhere. 2,500 serotype, Salmonella enterica and Salmonella bongori. And I can tell you that the majority of virulent or pathogenic Salmonella are from Salmonella enterica. We classify Salmonella, so we serotype Salmonella based on the antigen, the capsule, the O antigen or cell wall antigen, and then if they have an antigen for flagella. And I want to stop a second here because it's gonna come back later in my presentation, but if they lack the H antigen, they are classified as monophasic. And Raul already say that there is a lot of attention on this type of Salmonella monophasic because it is multi-drug resistant. And so we are trying to do everything to get rid of this stereotype, but it's a good example of microbial adaptation. Okay, when you get infected with salmonella, you get salmonellosis. It can take up to 48, so 2 days to show up, but then you get sick for almost 1 week, 1 to 2 weeks. The average dose that you get infected is 10 to the 4 CFU per gram of food, but depending on the virulence of the serotype. In the United States, we use a lot this term, YOPI, and that means young, old, pregnant, and immunocompromised. And like this, we classify most of the time that the population that is target of this type of infection. In case you don't remember, but when you get salmonellosis, this is a foodborne pathogen, so that means you need to eat an infected food. And then salmonella will arrive to the intestinal epithelial cell, will be captured, and then it's gonna multiply in the vehicle. It's gonna lyse, and it's gonna use a type 3 secretion system, and it's gonna go to the bloodstream and then cause infection. Going back on how everything started in my lab, in 2012, the Food and Drug Administration in the United States did a survey of all the serotype of salmonella that were present in animal feed. The Food and Drug Administration in the United States regulates animal food as well as human food. Not all categories they share with the USDA, but a lot of human and animal food is regulated by FDA. And so they went around this feed mill facility, and they rank the top 10 serovar that they were able to find in feed. And I want to point your attention on number 5, and number 6, that are Salmonella typhimurium and Salmonella monophasic. And so around the same years, they also published, another group, they published a study that ranked the number and incidence of Salmonella infection for human. And Salmonella typhimurium and Salmonella monophasic come back up again in the top 10. So our question was, after seeing this surveillance data, is there a connection between feed and feed mill as a potential entry route of Salmonella in the food supply chain? And yesterday, when we talk about welfare and sustainability, you guys understood that we are part of a continuum, right? And so everything is related. Now, a disclosure. Pig farm in the United States are very different than poultry farm. Poultry farm are vertically integrated, so you are able to understand what really happened from feed to slaughterhouse. Why the pork supply chain is a little bit different. That means that you will have different facility that produce feed that will help different farm. And sometimes it's by region, and they might not be connected. So this surveillance is difficult. So the first thing that we did in my lab was, is it really true that we can find Salmonella in feed? And then if we find Salmonella, let's try to characterize them from the genetic and phenotypic point of view and understand if we if they are antimicrobial resistance, and if we can find some gene that confer heavy metal resistance, because sometimes heavy metal are used in feed production. Okay, so what we did, we selected 10 main sites in the United States, and we select them based on their production in the pork supply chain. We visit this facility 3 times in 1 year, and we selected 12 sampling sites. Going back to what Raul said, we did not only sample environment or feed, we also sample people, so the boots and the broom. We follow an USDA FSIS method of salmonella from the environment, and then we use phenotypic method, but also we confirm them by PCR. Once we found salmonella, confirmed by PCR, we did whole genome sequencing, and we evaluate pathogenicity island, virulence, antimicrobial gene, presence of heavy metal genes. But then sometimes bacteria can have gene, but they can decide not to express them because of the pressure of the environment. And sometimes happen the contrary. Sometimes they show the resistance, but they lack of gene. So what we did was we double-check what we found at molecular level with an antibiotic resistant test. And we also, that was just a sensitizer. So it's a plate, like a 96 well plate, where there are all the antibiotic that you can think about. Then you put your strain, and there are going to be a level of optical density that is going to help you understand if the gene is expressed or not from the phenotypical point of view. And then we did a agar dilution method to verify metal resistance. You guys probably use a similar method for antibiotic resistance. So we are looking at the ELO. Okay. Oh yeah. In case you are not familiar with feed mill production facility, Luciano might be, because in the state of Paraná there is a lot of feed mill production, very similar to food processing production. So the ingredients are going to come. They're going to be a storage area, but also a manufacturing area. And then the difference is the feed is going to stay in these beans for a relatively long time, and that's kind of the problem, because they're gonna wait the truck that is coming, and that they're gonna open up, and then the feed is gonna go inside the truck and then go to the farm. And so depending on if you are collecting feed for poultry or for swine, you might have some feed staying here for a couple of days, while they do a relatively good job in cleaning the storage and manufacturing area. It's almost impossible to clean these bean boots. They are huge. There is no way that they can clean, and you will see in my data that this is the point of contamination. Okay, some results. So you can see that these are the PCR confirmed, and we collected almost 400 samples, including environmental and feed, and only 17% were salmonella positive. We have higher salmonella positive at the beginning of the process, so when the ingredients come. Then when we are in the production area, you can see a decrease. But then again, at loadout, agar, and the finishing bean boots, you can see that there is back an increase. I also wanted to show you that the broom and the shoes have relatively high PCR positive. So they use the broom almost anywhere. They don't care warehouse, production, and so this is a good vehicle of transmission. We saw that depending on the season that we were going to sample, so when it was warmer, we found more salmonella, not surprising. The sampling site was significant. I told you that at the beginning of the cycle, we see higher salmonella, then manufacturing the decrease, and then the bean boots, we increase again. There was no difference between if we were going to sample a pelleting mill, so where they do have a heating step, or if they did not have a heating step, and that was interesting for us because we were thinking that the heating step, yes, it kills salmonella, but then when they store the feed that is ready, that's when it gets cross-contaminated again. And this is what we mean for meal ID is if the first time we went to a meal, we saw high presence of salmonella, every time we were going, that meal was always full of salmonella. So, like sometimes we say listeria is persistent and you cannot get rid of listeria if it's entering your dairy production. It seems salmonella is very similar. Okay, sorry, I don't know if you see, but this is kind of an overall again of the significance of the season. Fall and summer were warmer than spring. And then if you look at this Kansas meal, all the time we went was full of salmonella. Same thing from North Carolina, except in the spring season when it was colder. If you look at this type of serovar that we found, you can see that there are a lot of serovar, monophasic, ricin, typhimiorum. All these serotypes, they are also present in salmonellosis human infection. So they don't care what is there, but I think we should start caring because there is a potential for human infection. When we start playing with bioinformatics, these isolates were divided in 14 clusters. And every time we were going to look what was in the cluster, what was in the tree, you can see that the majority of the other isolates that belonged to that cluster were clinical. And so that means that there was a connection between our environmental sample or our feed sample with other people that got sick with an isolate of salmonella that is genetically related. When we instead correlate what we found in our model for the phenotypic and genotypic comparison of antimicrobial resistance and heavy metal resistance, we found that our model was 99% accurate. We found antimicrobial resistant gene in 51% of our salmonella. 58% have at least 1 antibiotic resistance. These are the most common antibiotic that we found. We find a lot of metal resistance for copper, mercury, and silver. And most of the time, when we found an isolate that was resistant to metal, was also resistant to antibiotic. And this is pretty common because, again, the environment put pressure against bacteria, and if they develop a resistance for antimicrobial, antibiotic, they will also develop a resistance for metal. Everything that in the environment can be detrimental to them. We also did a questionnaire in those facilities because we were trying to understand it's because they are old, it's because they are getting ingredients from other countries, it's because they are small, they are big. We even asked them if they do some microbial testing. They don't care about pathogen. They do not have resources or interest to do any micro data, and we couldn't find any significant factor in supplier or volume, or if they were only for swine, the age of the facility. We think because maybe our sample size was relatively small, we couldn't find anything that was really significant. So, for these two studies, we concluded that our isolate, environmental and feed, were clustering with other isolate that were coming from pet food, pork, and clinical sample. The serotype that we found were pathogenic for human, but also for animal, so they can constitute a public health concern. There was, in our model, a strong agreement within predicted and measured resistance, and we found a lot of multi-drug resistance serotype. We concluded this study and say, okay, we cannot exclude the risk of salmonella from feed. I think it's important to consider feed as an entry route of salmonella in the pork production chain. So we could end here, but then we decided to do something else. And so while we demonstrate that there is a risk, we wanted to say, okay, is a significant risk. The pig is eating a contaminated feed or is drinking contaminated water or is getting salmonella from air. Do we find this salmonella in the pig itself when we go and sacrifice the pig? So, going back to why we choose monophasic, I think Salmonella monophasic show up in the 80s in Portugal from chicken and then in poultry. There was no Salmonella monophasic in the United States until the 90s. We think that because we import and export food from all over the world, Salmonella monophasic migrated from Europe to America, and now is almost one of the most common salmonellosis, Salmonella serotype that cause foodborne outbreak. It's coming up continuously in the United States in pork, and very recently we got a recall and outbreak where the leg of the pig was identified as a source of the outbreak. So what we did, this was a CEAD grant. So was not a huge grant, was just a proof of concept. We only had 12 pigs, approximately 3 weeks old. We have a facility at Kansas State University where we can infect animals with foodborne pathogen or viruses. So we selected 4 rooms. In one room, these pigs were not treated, so they represented our control. In another room, we gave them contaminated feed only on day 1. In another room, so pens, they were separate, so there was no risk of cross-contamination between treatment. We gave water. And in another room, we made an aerosol. I will show you in a second picture. Okay, so this one was easy to give them contaminated feed. We prepare a slurry. We opened the mouth of the piggy. They were eating. We needed to do a gavage for the water. Because they had the feeling of rejecting it. So we needed to make sure all the 10 ml of infected water went down. And then, I don't know if you can see, but you know the aerosol for the kiddos? Same thing we did we try a lot of things, this was the best. Sometimes salmonella can be in the air, so. That's the 3rd treatment that we did. Okay, and then we just did the treatment on day 1, and then we keep them separate, but we treat them normally. We give them food, water, we weighted them every day. We collected fecal sample from the floor, we collected rectal sample. By the end of this study, this pig, as soon as we were entering the room, they were just pooping because they were scared, they didn't like it, the stimulation. We got nasal swab. We checked the floor, we checked the feeder, and we checked where we were giving them water. And then after 1 week, we sacrificed them and we checked the presence of Salmonella monophasic in lung, in some lymph nodes, in the digestive tract, and then we were very interested in understanding what was happening in the leg. Okay, so in order to make sure we were finding the same salmonella that we put them, we use a method, a kind of a sandwich method. So we process our sample and we plate them in EB Petri film, that is Enterobacteriaceae Petri film, but then we transfer the Petri film on a XLD plate, and then we count only black colony that are salmonella, and then we confirm the PCR with, so we confirm salmonella monophasic with the PCR. And I think, Raul, we use a method that you cite, Barco, is one of the few that did the multiplex PCR on salmonella monophasic. So, and this is how we analyze them. Our fixed effect were the treatment, the day, and then the interaction between treatment and day. Okay, so what we found when we were looking at the feces, not at the rectal swab, so feces, what we were found on the floor, the pig infected with, just beautiful, by the way, I was surprised, my student was so good. No salmonella at all in the control pig ever. I don't know how she did it, but she made it. So, no salmonella in the control. We found salmonella on the feces on the floor, mainly in the pig that were infected with contaminated feed and contaminated water. I can tell you that at day 7, we probably find salmonella in the environment with the aerosol because they were starting to have diarrhea. And so that's why we found it in the feces on the floor. Let's see. Okay. Rectal swab, so no contact with the environment directly from their body, kind of same thing. Only pig infected with contaminated feed and contaminated water did have rectal swab positive. We weren't able to find rectal swab positive for pig infected with aerosol. And that's maybe also because of the number of our sample. Maybe if we did increase the number, we would find different results, but that's kind of the limitation of this study, but was a kind of proof of concept. But makes sense, because when we look at the nasal swab, the pig that were infected with aerosol are always positive. And then the pig infected with feed were also positive. What's happening, if you remember one of the first slide that I show you, the fecal sample from the feed pig are always positive in the environment. And the pig are touching fecal sample and the environment with their nose. And so that's an explanation of why the feed pig might have the nose positive. And when we talk about environmental sample, again, feed and water are the ones that show up for the positive Salmonella monophasic. And I apologize, but this is just a summary of the results that I show you. And you can see we couldn't find anything in the control. When we look at the aerosol, mainly in the nasal, and then instead the pig contaminated by feed and water, kind of all over the place. Yeah. After 7 days, we sacrifice these pigs, and we look for the presence of salmonella in the lung, in the lymph node, in the digestive tract, and in the leg muscle. And I can tell you that every time we sacrifice the pig that were contaminated by aerosol, we were able to see a positive lung. We also saw the tonsil positive. Across the board, when we look at the lymph node of the jejunal, aerosol, feed, and water were contaminated. Tonsil also all across the board, the intestinal tract, aerosol, feed, and water, they were all positive. And also some part of the ileum. When we look at the muscle, none of them were positive. And so this is telling me that, like Raul was trying to explain, if the lymph node, if the digestive tract is contaminated during processing, there is a huge risk of cross-contamination. I asked some help to my vet friend, and we wanted to do some micrograph of the tissue that we analyzed. And so this is a muscle where there was no salmonella, and so you don't see any lesion. But then instead, this is a cecum from a pig that was infected with a feed. And you can see some epithelial damage and you can see here some neutrophil and debris. So it looks very different than the non-lesion tissue. And so, conclusion, I think Salmonella is a risk. I think we should do something. United States is working towards it. I think feed is an underestimating way to carry Salmonella in the food production chain. We show that if the pig gets Salmonella from feed or from water or from aerosol, we can find in the digestive tract and in some lymph node, and this is pose a high risk when we process our meat. And thank you. And question? In a little bit.
It's working now. So I would like to invite Valentina, Paul, and Raul to the round table. And we can start the questions. So now we can start the discussion session. And anyone, any question for the guests? No one? Oh, we have a question. No, you can start.
First of all, thank you to the three of you. As you know well, I'm a field person, not a lab one, so something is more clear now to me. Just for each of you is the same question, because I would like to better understand what these students can do once they will be in the work field to help decreasing the importance of the two, because one was Salmonella, pathogens you spoke about. So some practical advices once they will be in the field and maybe helping in a nutrition company or as a vet or as counselor for farming. So there is something they can bring for all this in the field to better help the farmers and the productive chain in a very practical and easy way for the students.
Yeah, so thank you for your question. I think the last slide of Raul, I agree with him and kind of summarize what we can do or what student or the next workforce can do. I think the approach in Europe and in Brazil and maybe in United States is a little bit different, but trying to add some additive or antimicrobial in the feed or in the water, and definitely increase the sanitation system might help, in my opinion.
Well, I will add that perhaps the most important thing is to be aware of this. I mean, it's because nobody cares, but there is a clear association between Salmonella in animals and people. And we know, now we are in the One Health thing, you know, and this must be said. I mean, we have to do something. If they know that they have to do something in each of the places they are going to work, because there will be people working for the government, they will be to translate to the government that this is an important thing in public health. There will be people working in the feed industry. There will be to transmit that the feed industry has a role in there. The farmers, the slaughterhouses. I mean, it's information first, and then look for the tools. And actually, what we are doing in Spain is to show some tools that people can use. But in the future, when they decide that they have to do something, because so far nobody's doing anything.
Okay, so I'm going to talk from a diagnostic perspective, obviously. So I've been asked a few times by actually vets in Europe, so I'm not sure how this applies to Brazil, is if you have all these good diagnostics, is this going to put me out of a job? Yeah, so actually, no. So I would say that you've got to approach with the diagnostics and say, okay, I need to understand how those diagnostics were derived from, I need to understand the sensitivity and specificity, but ultimately, and I said this at the beginning of my talk, they're an aid. You have to have the whole picture, what's happening with the herd, maybe other tests, all that kind of stuff. So you've got to, it's part of the puzzle, it's part of the diagnosis, if you like, how you're going to take things on. But do understand how those diagnostics were, don't just say, okay, it's come from this company, it's positive and negative. Understand how those diagnostics were derived. Understand the positives and negatives of those weaknesses, etc., of those diagnostics, and put it in the overall picture. And from what I've seen, sometimes that gets forgotten. So if you remember nothing else from what I've said today, that is a good message.
Any questions?
Hi. First, good morning for everyone, and I'd like to thank all of you for your presentation. And my question is for Dr. Paul. It's two questions, actually. First, do you think that the Brexit had some sort of accelerated the development of diagnosis tests in the UK? Because considering the cost of transportation to Germany and other countries, it's a major cost for diagnosis. And do you think the Brexit has caused this to become more important for the country? And second, do you think that it's feasible to develop a CRISPR-Cas12 diagnosis test to become part of the routine for a lab, like here in the Department of Veterinary?
Okay, can you repeat the second one for me? I missed the first bit of that.
Do you think it's...
sorry, the second bit of that. So
it's feasible to develop a CRISPR-Cas12 test based in, to become part of the routine of a lab, such here in the Department of Veterinary, or to become a commercial test for a lab, sort of?
Okay, so the first one regarding Brexit, that's very easy, no. So I think, okay, I didn't vote for Brexit. I'm still getting over this, but never mind. But no, I think that the applications will happen anyway. Is that, yes, if you take our case, we went for a UK-centric initially because we're having to test in the UK. But actually, we've done this globally. So we try to think globally, and the tests that we've developed could be used globally, for sure. So definitely the answer is no for that one. So CRISPR-Cas, so I've been involved in studies with that, so as well, with some Chinese collaborators, so with Strep suis, and there's been quite a few papers on this kind of stuff. So what's not happened that I'm aware of is it's got to the stage where there are tests in the clinic. Can you make tests based on that? Yes, from my own experience, I'm on a few papers with CRISPR-Cas for that. So yes, you can. But can you develop a commercial product? That will happen. Something will win out, but I can't give you a timescale on that. But I think the answer is yes. But is CRISPR-Cas the best way of doing this? I don't know. Which platform will win out is always a thing with diagnostics. You never know. And the big companies have to be happy with it, essentially. And they will determine it eventually. I hope that's answered your questions. Yeah.
Questions?
Thank you very much. All these presentations was really wonderful. So I have a question to Valentina first. So you mentioned about these heavy metal resistant genes present in Salmonella species. So I was wondering, did you look specifically which antibiotic resistance was related to, the presence of heavy metal resistant genes? Specifically, you mentioned about copper and zinc, those metal resistance. So is there any relationship you got with any specific antibiotic resistance or maybe antibiotic resistant genes?
Yes, we did. I don't know on top of my head, but I can get back to you with that. Yes, we saw some relation with always the same, or not always, most of the time the same antibiotic and the heavy metal. So I can get back to you with that.
That would be interesting. Yeah.
I think there has been a decrease on the use of heavy metal in the United States in feed. But still, I think we bring back the previous usage. So I think since instead we are continuously using antibiotic, the same gene are always there, and based on what they are stressed with, they will show.
And is there any control measures by some authorizations like FDA? Do they monitor the uses of particular heavy metals in feed?
FDA does.
Does?
Yes.
Okay. Okay.
Yes.
Good morning, everyone. Thank you all for the great presentations. So my question is for Dr. Raul. What some countries of European Union did to eliminate the salmonella of their countries? So my research, the objective of my research is reduce to use of antimicrobials in the swine production. For this, we are selecting bacteria as select with probiotic potential and activity against salmonella. So I think is an area that we can develop more solutions.
Bueno, I agree. There are many other options. Now, yeah. Antibiotics cannot be used to get rid of salmonella. I mean, they are not used because you will be using an antibiotic to prevent something that is not producing a disease. Antibiotics in Europe are only used in case of disease. So, although you know that there are salmonella in your farm, you can use antibiotics there. Salmonella is a bacteria that is able to develop resistance very easily, actually. And the levels of resistance in salmonella now are very high in general, right? So just forget about antibiotics. So the next options are prebiotics. We did a study with a prebiotic that worked very well, but we used that for the whole growing finishing period. And it's quite expensive because it's based on a plant that I don't know how, I mean, it's completely different than here. It's European tree, and the fruit from that tree, you can get some powder that you can use. It's good for humans and it's good for animals. And because humans use a lot, the price is usually high. And because of the climate situation, the harvest change from one year to another. So it's a kind of costly product. So difficult to use in livestock for a long time. But it's working very well. And probiotics, there has been some studies working with probiotics, mostly on piglets. Most of the studies has been done on piglets because it's when they change from the lactating period to the nursery period, in which they change the diet. There is a lot of problems, digestive problems there, and many bacteria, E. coli, Salmonella, may survive and give some problems. So they are trying some probiotics in there. I think that there are many different results, because the thing is look for the good cocktail, because usually it's not just one bacteria, but a cocktail of bacteria that will work better, right? Same with organic cases. There are a lot of organic cases, a lot. And the problem is when you should give them, for how long, which dose, what kind of organic cases, a mix of organic cases, just one. There are a lot of things that has to be decided, and there are a lot of tests. Some went well, some went wrong, they didn't get anything. So the problem is to find, and the major problem to control Salmonella in the pig farm now is to determine when start the risk. I mean, when the infection is starting in the farm. And I think that we should look not just to the feed, that as well, but to the sows. We have to start with the sows. We have to clean the sows to have litters clean of Salmonella, and then from there try to keep them clean. So there are a lot of treatments that are possible, but we have to still working more on that. So what you are doing is perfect. I mean, you have to continue with doing that and try to find the doses, the bacteria, the time, exactly, all these kind of things, yeah.
Any more questions? Anyone? We have some minutes yet? No? Any questions in the virtual mode? No? So, okay, so I appreciate our lecturers, and I want to say thank you to Dr. Paul, Dr. Raul, Dr. Valentina, and we can finish today, and we will continue tomorrow, okay? Thank you, everybody, to coming, and see you tomorrow.